far, we have talked about biogeography and the fossil record as evidence for evolution. So today we are going to take a look specifically at number 3 and number 4, and we are going to talk a little bit about anatomical and developmental similarities, and we are going to talk a little bit about molecular biology. We are going to save living populations for a little bit later in our series in Unit 8. So let's begin by talking a little bit about anatomical and developmental similarities. So one of the things that scientists look at as evidence for evolution is in comparative anatomy. And this is basically where we take a look at different organisms, and we're going to take a look at the specific fossils or the bones of different organisms. So in the case that you see here, we're looking at a human arm, a cat leg, we have a bat wing, and a dolphin flipper. Now these colors here are actually very important to recognize because we have the similar bones in all of these 4 organisms. So we have our radius and our ulna, we have in our human arm, we also see that in the cat leg, the dolphin, and the bat. We have our carpals, which are our wrist bones, and we have our metacarpals and phalanges as well. And we also see those in different forms in the other 3 organisms. So it's possible that we can compare different organisms and take a look at the different bones and possibly kind of see if there's a common ancestor here. So the 2 words that we kind of take a look at when we are talking about comparative anatomy is a homologous structure, which is a structure that have common structure but may not share a common function. And an example of that would be the wing of a bat, like I have here, and the forearm of a human. So I just talked a little bit about that and how we see the same bones. So we have very similar structures here. We just have very different functions. The arm, we carry things with our arms, we can grasp things with our hands, whereas a wing, that's definitely for flying purposes in a bat. We also see analogous structures, and these are structures that have a common function, but not a common structure. So an example here would be the wing of a bee versus the wing of a bird. Both of them fly, so that's where we come up with the common function. But the structure of a wing of a bee and the wing of a bird are definitely not similar to each other. Another type of organ or structure that we can look at is something called a vestigial structure. Now, these are structures inherited from ancestors, but they've lost a lot of their original function due to different selection pressures in the environment. So, for example, the hip bones or the pelvis of a whale is a vestigial structure. We know that whales have come from land mammals at one time. So whales were able, not specifically whales, but their ancestors were able to walk on land. Their vestigial structure is the pelvis bone. There's really no reason for them to have this bone unless it's a vestigial structure, meaning that an ancestor at one time had the means to walk. In humans, we actually have a few different types of vestigial structures. The first, of course, are our wisdom teeth. We actually have very little room in our mouth anymore to have our 3rd molars, which we call our wisdom teeth. And way back in the day, before agriculture, we actually needed these for our diet in order to eat the raw vegetation that we were eating. We also have ear muscles around our ears, and these are to move our ears. Now, we see this a lot in other organisms like dogs, cats, even, lions, tigers. They're able to move their ears in order to locate sound a little bit better. We still have those muscles, but we really don't have the ability anymore to move our ears. We really don't need them. So we don't really need to follow the sound. We'll just move our head if we need to. We also have a coccyx bone, and this is our tailbone. So as you'll see in a few slides from now, in our embryo stage, we actually do have a tail. So this is just the remnants of that tail pre-birth, basically. And we also have an appendix. which is located right off of your intestines, and this is thought to have some sort of digestion properties. We can live without an appendix now. We really don't need it. In fact, if it ruptures, it can cause a lot of issues for us. So we've discovered we really don't need the appendix anymore. So vestigial structures are just those that have been inherited from ancestors, but pretty much have lost their function. Now, as I mentioned in that last slide, humans have a coccyx bone, which is that tailbone. Well, we don't have a tail anymore, but this is because when we were an embryo, we actually did have a tail. And there's a certain science where people study embryology. And this is the study of the development of an embryo from fertilization to fetus. And embryologists typically might study one type of animal or organism, or they might study a whole gamut of them and do evolutionary embryology. We have a picture here that, of course, if you look down on the bottom, you'll probably see what we are. But if you just cover your hand a little bit and just take a look at this very top section here, the one that I'm going to circle really horribly right there, everything looks the same. Just by looking at this top row, you probably cannot determine what is the human and what is anything else. So before DNA studies really came about, early researchers and early embryologists noticed that there are these similarities in the developmental stages of many vertebrates. Like I mentioned, we all look pretty much the same. Now, as we have thrown DNA in the mix, we have genes now, and we've confirmed that the same groups of embryonic cells develop in the same order and in similar patterns to produce similar tissues and organs in vertebrates. So if you take a look at a lot of our vertebrates that we have, most of them have hearts, we have brains, we have a digestive system of some kind. There's very similar tissues and organs in all our vertebrates. So once we're a fetus and an embryo, we kind of start to change a little bit into what we are now. But you'd kind of never guess just by that first stage right up top. And that's what embryology is. It's the study of these embryos. And like I said, notice we have that tail, but we sort of lose it a little bit later along as we go through our trimesters. Now, I started to kind of talk a little bit about how DNA and genes came about, and we've really utilized those studies in order to help us understand a little bit more about evolution. And so our fourth piece of evidence of evolution is molecular biology. So molecular biology here, we're taking a look at number 4. Now we do know after several studies of our DNA, along with other organisms' DNA, all organisms have the same molecules that make up DNA. We've discussed those in earlier videos. We're all made up of adenine, thymine, guanine, cytosine. We might have different orders, but we're all made up of those. This provides us a universal genetic code. All organisms have the same stuff. Again, just organized in a very different way. Now, because of mutations and reshuffling of genes during sexual reproduction, we have a certain amount of variation on which natural selection operates. We'll talk more about natural selection soon when we start to talk about living populations or even fossilized versions of evolution. But just know that a lot of these mutations and reshuffling of the genes are really the causes for the changes that we see over time. Now, something that we've already talked about here is this process of transcription and translation. And actually, this is incorrect here. This should be transcription, and that should be translation. But we've talked a little bit about how this process happens, and we've talked about how mutations can happen when DNA is replicated in the transcription of the RNA and in the creation or that translation into a protein. So we can see mutations along the way here. One of the things that have kind of revealed also pretty recently itself is the cytochrome C amino acid and are basically the evolution of the different number of amino acids that are different between different organisms. And as you can kind of see, between a chimpanzee and a human, we actually see 0 differences in the amino acid in this particular cytochrome C gene area. A rhesus monkey, we only have 1. A rabbit, we have 9. If you look all the way down at the bottom, a yeast, we actually have 42 differences between a yeast and a human. So that kind of gives you an idea of just how different that we can be from a yeast. But cytochrome C evolution has seen a big step since we've kind of discovered this. And when I say recently, I'm talking 10, 20 years. So we can sequence homologous stretches of DNA from different species and use them to test for relatedness. And that's kind of how they came up with that cytochrome C evolution here. We're taking a look at the similar sequences amongst different organisms. One of those homologous stretches that we look at is something called the Hox gene. Now, the Hox gene controls development of the head to the tail body axis in animals. So here we actually have a human, and we have a mouse embryo here. So they are a little bit different, but the Hox gene is found in almost all multicellular animals, from fruit flies to humans. So Hox genes probably are one of those earliest types of stretches of DNA that helped create evolutionarily some of these different kinds of animals. Now, if you take a look, we see the difference here in some of the differences of our body type and structure. So we have Hox genes numbers 1 through 4 take care of the head in humans. Now they're different colors, so we can't compare them exactly. But if you notice that 1 through 4 in the mouse also takes care of the head. Now 5 through 6 here takes care of the neck. 5 through 6 here also takes care of the neck region in the mouse. And so on and so forth. Number 7 and 8 in the human takes care of the trunk of our body. Now 8 is missing here for Hox A, but we have this blue version over here, so that takes care of the trunk of the mouse. This just means that in mice, they don't have that particular Hox gene. Now this one continues on from 9 to 13, and this is basically the limbs of a human, and 9 through 13 in the mouse. Here we can't really tell, but as you see, it's possibly that this also are the appendages in the mouse. So the Hox genes are very similar in multicellular animals. They're seen in many, many different types of animals, fruit flies to humans. And yes, fruit flies are animals. Now, these changes in DNA might cause changes in development. So as we were talking about how proteins are created in protein synthesis, we have one slight mutation that could change an entire sequence, for good or for bad, but the whole sequence gets changed. Now, if something happens where it happens to be an advantageous change or a mutation, then natural selection is going to kind of act upon that. And those particular organisms that have that mutation will start to see a rise in the number of organisms, the number of individuals that have them in a particular population. So for example, let's take a look at this 3-spine stickleback that I have here. Now this top picture, we can actually see these spines. Now these are kind of like really sharp daggers that they have. And these were actually developed in defense against predation from fish. So basically when a fish were to bite down on it, it would get stuck and then it would release the fish. So it's a defense mechanism basically. Now, over time, though, there's been these mutations in this particular gene, the PITX1 gene. Now, this particular gene, so this one right here, the PITX1, this particular mutation actually changed the way that these spines were developed, and they lost their spines. The mutation was for a loss of spines. Now, what happened was that it actually was discovered that by these individuals that there was a pressure for these particular spines being lost, where it actually helped them escape crustaceans that lived on the waterbed. So any type of crustaceans that were going after these particular fish, by not having the spines, actually was more advantageous for them to get away from their predators. So over time, those particular ones that did not have the spines, those ones were able to reproduce, and those ones were able to then populate this particular area of sticklebacks. So natural selection will act upon the trait that is most advantageous for that organism, that population to survive. Now, I mentioned living populations, and I'm going to touch on it a little bit here as the fifth piece of evidence for evolution. Now, living populations we'll discuss more when we talk a little bit about natural selection in some future videos. But Darwin, other researchers actually observed natural selection within living populations, and we can see that today. We have basically we have a lot of resistance by bacteria to our antibiotics, and this is not a good thing. Because as bacteria create a resistance to our antibiotics, that just means that more bacteria are able to reproduce. And they're able to reproduce with that particular resistance. This is what's causing MRSA or some of the other types of really horrible bacterial infections that we see in hospitals or just in general. They're becoming resistant to antibiotics. That's an evolutionary change. And for bacteria, this happens fairly quickly because they are able to reproduce very fast. We have seen this also in the peppered moth, which we'll talk a little bit more about in future videos. But during the Industrial Revolution, it was actually more advantageous for the black moth here to hide itself among the smokestacks and the trees and all those things that had a soot on them. And these poor white ones actually stood out, so they were actually eaten a lot faster than the black ones. When the Industrial Revolution started to change a little bit, where we didn't have soot covering everything, then the black moths were the ones that stood out, and so those were eaten by predators more so than the white ones. We'll be talking a little bit more about this later. And another one, of course, I mentioned in an earlier video are the finches of the Galapagos Islands and their various number of adaptations to the foods that they eat. These are all things that we've seen within lifetimes, so within a 30-year span. And I would be remiss to not discuss human evolution, which again, we'll probably do more in class, but this kind of gives you an idea of some of those major changes that make us human. So there's about five, definitely a lot more, but five characteristics that are very important to understand on how humans evolved through time. Now there's some major advances that have kind of taken over, that have humans have done over the course of about 6 to 8 million years. So it's been a long time, but basically within the past 10,000 years, we've discovered agriculture. Humans themselves, modern day humans, have really only been around for about 250,000 years. So it hasn't really been a whole lot of time. There's been many other human or humanoid species before us. So we are not the first. Will we be the last? Who knows? Some of the major changes that have made us who we are. Definitely bipedalism. You really don't see a whole lot of other animals out there that are bipedal, walking on two legs. We have an increased brain size, and we're actually not the the human with the largest brain, that was, I believe it was Neanderthalensis or the Neanderthals, but they had a larger head than we did. Increased jaw size. Again, we're not the humans that have the largest, but we also we've been developing a smaller and smaller jaw, which I mentioned earlier with the wisdom teeth. Language acquisition. Humans do have our own language, although other organisms do have their own way of communication, but we definitely have language. And tool manufacturing. We are, as far as we know, one of the only types of animals that can actually manufacture tools. Although this particular one has been in question recently, where we've discovered some different types of chimpanzees turning sticks into tools, certain other types of monkeys using rocks and making their own tools out of rocks. So this one comes into questions a lot. We'll talk more about human evolution in class, but for now, this is a good way to understand the 5 main points of human evolution.